Okay. Shall we? Okay, so we're in. Last time we made a start with the parade of the elements. Shh. We're in the middle of a parade of the elements. Last element you saw was copper, and the next one is zinc. These are metals. They're part of the micronutrients. They're needed at low levels by plants. And they often function as cofactors for certain specific enzymes. Here we have another metal, zinc. And it also acts as an enzyme activator, particularly, basically two main functions. One is hormone regulation, and particularly the hormone auxin, which you will see later when we look at hormones. And one of the functions of auxin is to allow cell expansion to happen, so growth, basically. And therefore, a symptom of zinc deficiency is very short internodes, because that's where cell elongation often happens. That's where plants grow, in between the nodes. And you can also have smaller leaves, which also has to do with cell expansion. The leaves cannot expand. And there are also certain transcription factors, remember those guys, that induce gene expression of a bunch of genes. There are these so-called zinc finger proteins that hold zinc, and that's needed for their function as transcription factors. So those are some of the functions of zinc. And as you see in this picture, you see deficiency symptoms here in younger leaves, suggesting it is not very mobile in the phloem. The form of zinc present in the environment is mostly as the cation zinc 2+, and there's only that one oxidation state. So it isn't like copper and iron, a metal that can switch between oxidation states. It's just always zinc 2+. Next element, manganese, another metal, present in the environment as Mn2+, so again, just the one cationic form. Again, it's an enzyme activator for certain enzymes. And one of the functions of manganese is also in photosynthesis, particularly the part where water is split into oxygen and hydrogen. And manganese, there's a water splitting enzyme, as it's called, or sometimes called oxygen evolving complex. This has manganese in it, and it needs manganese to function, so that's why manganese is important for photosynthesis. And deficiency symptoms, you see here some examples in different plant species in the interveinal regions, and you may see them as chlorosis or as kind of gray spots, which is, the gray spots is kind of different from what you've seen before. Interveinal chlorosis is something you see also for other elements. So here are some examples in different species. And the manganese can be limiting from, I think, more at high pH and low pH. And so if that was in the slides as low pH, but I think that should be high pH. So I took it out in this updated version. I'll show you later a figure which shows bioavailability as a function of pH, and then we can come back to this. So we are almost there, molybdenum. Now we're getting into the regions of elements that are needed in such low levels that people actually didn't even realize that these were essential elements for the longest time. Just a few ppm is enough, and typically the plant can get that from the seed. So if you want to induce deficiency, you have to give them over several generations a lack of molybdenum in this case. And the other element is nickel, which is also in the low regions of need for the plant. The molybdenum is present as an anion, an oxyanion, MoO4 2- in the environment. Molybdate is the term. So it's a bit similar to sulfate, but molybdenum. Molybdenum is a cofactor of just a few proteins, namely enzymes involved in nitrogen metabolism, in assimilating inorganic nitrogen to organic nitrogen. And there's a special class of plants, the legumes, which have symbiosis with microbes that live in their root nodules. These are the legume plants, the bean family. So these microbes that live in their root nodules have a special additional requirement for molybdenum. They need it for their nitrogen-fixing enzyme. So that's why legumes have a higher requirement for molybdenum than other plants. So they're kind of a special case. And they don't really need it for their own physiology, but their microbial body needs it. So that's why if you grow legumes, you may want to consider making sure they get enough molybdenum. Other plants, it shouldn't be a problem. And for molybdenum, which is what you also see for other anions, it tends to get more limiting at high pH. No, it gets more bioavailable at high pH. Anions, as a general rule, tend to get more bioavailable at higher pH, like 7.5, whereas cations tend to get more bioavailable at a lower pH. That has something to do with H plus and OH minus concentrations, which vary with the pH, obviously. And they can compete with those cations and anions for binding sites on the soil, so that affects bioavailability. I'll show you more about that in the next chapter. So at high pH, molybdenum it is actually is more bioavailable. So the deficiency is more likely to happen at a lower pH. Chlorine, that's another one in the lower regions. Well, actually, the levels of chlorine in the plant are not that low, but it's not likely to be limiting. Chlorine is present as Cl-, so chloride in the environment. And that is the form that plants take up, and it's also the form in which it stays in the plant. So it just stays as Cl-. It can get accumulated actually to quite high levels. And it's needed as an osmoregulator, similar to K+ that you saw earlier. So if plants want to move water by osmosis, they have to first move solutes, and then water will automatically follow by osmosis. And then the solutes of choice often are K+ and Cl-. So those often are moved on purpose, and then the water will follow. So the good thing about moving K+ and Cl- together is that you don't mess up the overall electric charge in a compartment, because you move a positive with and a negative at the same time. So that's one function, osmoregulation, moving water. And the other function is in photosynthesis. The process of splitting water also needs chloride ion. So that's another function. So chlorine is usually not limiting for plants. It's hard to induce chlorine deficiency, but if you manage to do it, then you will see these symptoms: reduced growth, wilting leaf tips. That's something. It's a little bit special compared to other deficiencies, and just chlorosis, which is not very specific. So that's chlorine, and then nickel. Finally, nickel is needed at really low levels, similar to molybdenum, ppm levels. It's a cofactor of only very few proteins, and similar to molybdenum, those are the ones involved in nitrogen. It's slightly different enzymes involved in nitrogen metabolism, namely nitrogen mobilization. So that's particularly important for seedlings, which have stored some nitrogen in the form of amino acids, proteins, and that then have to be remobilized to make other things. So that's in the seedling stage, and the plants especially need nickel for that purpose. It's hard to make plants nickel deficient. Often you would have to deprive them of nickel for multiple generations. And then what you are going to see is kind of the usual symptoms: chlorosis, necrosis, so not very specific. So that was the parade of the elements. And hopefully I wasn't too boring. But later, as we go look at these different processes that these elements are involved in, then hopefully you will kind of see them in action, kind of making it more interesting. But at least you've seen them now. I mentioned earlier, all elements that are essential show this kind of relationship of tissue concentration of the element with plant growth. We focused on this part of the curve. Where it is still going up, the element is limiting plant growth. So you can ameliorate plant growth by giving more of the element until you reach the critical concentration, which is the tissue level of an element where it's just getting to the point where it's enough. And then if you give more, so if the plant gets more in its tissue, it's in the adequate zone. The plant doesn't respond anymore in terms of more growth because something else is more limiting. And then for the micronutrients, if you keep going, then at some point you see that growth is actually going to go down if tissue levels keep going up. So then it becomes toxic. So metals particularly become toxic at higher levels. And it isn't defined here what the width of the adequate zone is, but it could be like a factor 10 or a factor 100. So 10-fold, 100-fold, may be the difference between, for micronutrients, between the deficiency zone and the toxic zone. So when you grow plants, you want to keep plants in this adequate zone, not too much, not too little. Similarly, plants want to be in that zone. So they have to have sensing mechanisms where they will sense, I'm now getting deficient for that element, I'm going to upregulate uptake of that element. And similarly, they must have also sensing mechanisms when there's too much of a certain element, and then it should kick in some metal tolerance mechanism, metal detoxification mechanisms. When we look in the section 3 at plant stress physiology, we'll come back to metal tolerance mechanisms. But just for now, realize that for micronutrients, there's a sweet spot where you have to be not too high, not too little. For macronutrients, you can hardly give them too much. If you go really to the 100 millimolar range, then you will get toxicity, but that's then more an osmotic effect than a direct toxicity effect of those elements in question. But the thing that micronutrients can do, the reason why they can be toxic, is because they may actually cause oxidative stress in cells, meaning they can kind of wreak havoc, taking electrons or giving electrons to molecules that can create toxic reactive oxygen species, free radicals, like an oxygen with an extra electron. Superoxide is very toxic. Hydrogen peroxide is another free radical, also called a reactive oxygen species. So if you have metals, too many metals, they kind of start to give or take electrons from these molecules. Also hydroxide, OH minus is good, but if you take an electron away, then you get a hydroxyl radical, OH, no charge. That's another free radical, free reactive oxygen species. So metals can introduce these free radicals, and they can damage DNA, damage proteins, damage lipids in membranes, and overall can cause death. So it's really bad. So that's a direct toxicity effect of metals, some of which are micronutrients. And then also what you can have as another type of toxicity is that these excesses of certain metals may actually induce a deficiency of other essential metals if they compete for the same transporter. We're going to have a look at transporters in the next module. So then what you're seeing as, let's say zinc toxicity might in fact be copper deficiency symptoms, because there's just less of another element. They compete for the same transporters. And similarly, once inside cells, they may also compete for the same enzymes. If an enzyme needs, let's say, copper as a cofactor, and then there's so much zinc in the cell, it can also bind to that protein, then it can take the place of the copper on the enzyme. As now the enzyme doesn't have its correct cofactor, it's not going to work. So that's another type of competition. So there can be competition for movement across membranes with using the same transporters between metals, or you can have competition for binding to a certain enzyme. So that's toxicity. And similar to the critical deficiency level, which was at 10% below the maximum growth, but at the kind of the left side, the low side, low end of the adequate zone, there's also distinguished a critical toxicity level, which is where growth is starting to get impeded. We're at the right side, the high side of the adequate zone, by 10%, and then we call that the critical toxicity level. Yeah?
Are the symptoms of different metal toxicities pretty general as far as...
The symptoms of the toxicities of metals are often the same as deficiency symptoms. So the chlorosis, stunted growth, necrosis, deformity. So it's the very same symptoms. So you would really have to do an elemental analysis to see what's going on here.
Okay. They relate a little bit to the more specific deficiency symptoms they might cause?
So the symptoms that you would see could either be an effect of a deficiency of another element, but could also be the free radicals can cause cell death, so that could cause necrosis. You could also have free radicals impairing photosynthesis, and then the pigment gets bleached, so then you get necrosis. So it's not an inability to make pigments, but the pigments are broken down because of toxicity. And so these critical toxicity levels and critical deficiency levels are expressed as a plant tissue concentration. So not so much the external concentration, but the internal concentration. So after seeing all these essential elements, there are a couple of elements that I just want to touch upon. So you're aware of this class of elements. They're called the beneficial elements. And here are the 4 that we know for sure are beneficial elements. In addition, there may be more. And actually, there may be quite a few more, but these are the most well-studied. So the first one is sodium. Sodium is actually an essential element for animals like us, but not for most plants, unless there's C4 and CAM plants, which you haven't heard about yet, but you will hear in the next section. They have a special kind of CO2 fixation, and a little bit of a pathway associated with that, and one of the enzymes in this pathway requires sodium. So that's why these plants with this kind of metabolism actually need sodium as an essential element. And sodium is called Na because it is called natrium in other languages, Latin. And it's present at high levels in soda, so that's why it's called sodium in English. So that's one beneficial element, but it is actually essential, but only for some plants. Next one is silicon, Si. That's present in high levels in sand. And it's not essential as far as we know for any plant species. However, plants grow better with silicon because it is present in cell walls and gives the plant sturdiness. And this is particularly needed for the sturdiness of grasses and scouring rush. So monocots, that they can have really high levels of silicon. And they can still survive without silicon, but in nature they would fall over really easily. So if you would have weeds or something, it would lodge very easily without enough silicon. And silicon may also have ecological importance as a deterrent of herbivory. So it's a secondary function. But so silicon is not technically an essential element because plants can survive without it in a greenhouse if you pamper them. Selenium is another beneficial element, which is essential for mammals, for instance, and many other organisms, but not for higher plants. The essentiality of selenium may have been lost in evolution. But selenium is still a beneficial element for plants. Many plant species have been shown to give a positive growth response if you give them a little bit of selenium, but it gets toxic at higher levels. And a class of plants that has a particularly big growth response to selenium are the so-called hyperaccumulator plants, which grow around here because we are in a selenium-rich area, and you can see them growing in nature here. And they can have a twofold higher biomass production if you give them some selenium. And they can accumulate up to 1% selenium. But they don't apparently need it. Nobody has shown a function. People have seen that they have better antioxidant capacity if you give them some selenium. So those genes involved in antioxidant responses are more induced by it. The last beneficial element that has been really shown to benefit plants is cobalt, which again is an essential element for other classes of organisms like animals and bacteria. And cobalt is not an essential element for plants. However, legumes really can benefit by feeding them cobalt. So they're the only plant class for which it's a beneficial element. They can really benefit from it because their rhizobium partner, which I mentioned earlier when I was talking about molybdenum, this rhizobial bacterial partner in the nodules of these plants, they need cobalt as a cofactor. So that's why for legumes you may want to give them some cobalt. And here below you see the periodic table of the elements with the essential elements in green. And they distinguish here C, O, and H as kind of a special class. They're the big three, but they're essential elements as well. And then the beneficial elements are shown here in yellow. And people have indication that there may be a suite of other beneficial elements all over the place, especially pretty heavy elements. They often show a small spike in growth induction at very low levels, and then that effect disappears. So how exactly this works is not known. But there may be more beneficial elements. But as far as we know, these are all the essential elements that you've seen. So that concludes Module 5. That's it for the parade of the elements. So what's next? What's left? We're going to have a look at how these elements move from the soil up into the plant across membranes. So we're going to have a good look at membrane transporters, different kinds of transporters. And then we're also going to have a look at some assimilation pathways. So first, solute transport. Starting at the base, the soil. So all these elements that you've seen, all except carbon and oxygen, plants derive from the soil. And not all plants grow in the same soil type. So depending on the soil type, these elements may either be present or absent, and they may be present at different concentrations if they're present. Also, environmental conditions may vary and may affect to what extent these elements, whether from the soil, go into solution and then are bioavailable to the plant. So the form of elements may also vary with environmental conditions. If you think of elements that can exist in more oxidized or reduced form, you can imagine that oxygen content can affect this in the soil. So environmental conditions can affect bioavailability, and then the soil determines whether it's there in the first place. So to what extent it's there to become bioavailable. So soil type influences mineral abundance and then also bioavailability. Depending on the soil type, also the soil can hold on to its minerals more jealously, like clay can hold on to minerals more, and sand really doesn't have a hold on minerals. So once minerals get deposited on sandy soils, it doesn't have much in the first place, but you can have some deposition from maybe decomposition of plant materials. So sand may gain some nutrients, but then they leach them really easily with rainstorms. So bioavailability is also affected by the soil type, clay versus sand. So if we look at a soil particle here with soil water and then a root hair, a cell, then you have all these minerals floating about in ionic forms, cations and anions. And the soil particle can have charges on it. Especially clay has charges on it. Sand really doesn't. It's just SiO2 without charges. But clay has charges, both positive and negative, but more negative. And also humus, which is decomposing organic material, like plant litter, also has binding sites, has charged groups on it that can bind minerals. So this is kind of a good thing, this binding of minerals to charged sites on soil and on organic matter. It's kind of a slow release fertilizer over time, so it can hold on to these minerals when it rains. But then if the plant is good enough, is strong enough to grab those particles and release them from the soil, from these charged groups, then it's a good source of minerals. So the soil type influences how much there is of a certain element. And as you saw earlier, soil can have different texture. Sand has the biggest particles, clay has the smallest particles, and silt is in between. And then there is also organic matter. I didn't talk about that earlier. So that is degrading organic material, mostly plant material. It's called humus. Typically, organic matter is just a few % of soil. And it's just being decomposed by organisms and also by just chemical, physical factors, like freezing, thawing, rain. But also there are small microarthropods who decompose plant material, and then there are bacteria and fungi which decompose this plant material. And as that happens, the minerals become available to the plants again. And that may be in organic and inorganic form. So these, the soil, with especially the clay and the humus, has these charged groups that can hold on to minerals by electrostatic interaction. As you see here, negative groups on the soil particle can attract positive ions, cations. And this is called cation exchange capacity, CEC. So soil provides minerals through weathering. So that means they're falling apart, the soil particles, and they're releasing these minerals as ions. And then once they're in solution, through charged groups, they can hold on again just loosely to these ions, and that's called, particularly it's abundant, is important for cations binding to negative groups, and that's called cation exchange capacity. So this is a form, kind of a slow release. It's a buffer, a storage form of soils for cations that plants can take away from those soil particles. They have evolved mechanisms to do that. So the higher the clay content and the higher the humus content, the higher the cation exchange capacity of the soil, which is a good thing, because that provides cations to the plants over time, and it holds on to these cations so that they don't wash out with the rain. Any comments about that? Yeah?
Like you said, the higher amount of clay present, that's the higher species.
Yes, I said the higher the clay content is, the higher the cation exchange capacity is. Similarly, the higher the organic matter content, the higher cation exchange capacity. Yeah. So, something to point out, I said plants have evolved mechanisms by which means they can make these cations available for themselves and take them from the soil. So the cation exchange sites on the soil particles bind cations, many of which are minerals that the plant needs, like you see here, potassium, calcium, copper, magnesium. You can fill in the rest. And so the mechanism that plants have evolved to make these available, they actually extrude protons. They pump out protons from their roots, so that's H+. H+ is positively charged, it's a cation, and it can replace these other nutrient cations, these minerals, from the soil particles. So they can replace, for instance, magnesium. Magnesium goes away, goes into solution, H+ takes its place. Now the magnesium can reach the root and can be taken up by the root. So they replace cations by means of H+, which they pump out of their roots. So the rhizosphere of plants, which means the root zone, just 1 millimeter around the roots, is acidified by the plant. They pump out H+. And this acidification, all these H+, make it possible for other cations to go into solution because they replace these cations on these cation exchange binding sites. Yeah, question? Yeah, the mechanism that is shown in this picture. So the question is, what about this CO2 being released? So this would be respiration releasing CO2, because respiration goes on in all cells, like we are breathing, plants are breathing. So that's another way in which plants can acidify the rhizosphere. That can react with water, and then you get H2CO3, which can then release protons as well. But they also have proton pumps in all their cells that pump protons continuously out of the cytosol into the cell wall, into the apoplast. So it's a combination of both of those things that acidifies the rhizosphere. So that's one mechanism by which plants are not sitting ducks, but they can actually scavenge minerals. They're not just waiting for them to accidentally bump into them. They actually go out and release them and go and find them. So as you can imagine, if you look at this picture, the more H+ you have in the root zone, the more bioavailable cations are going to be. And that's not only in the root zone, but anywhere in soil. So some soils are also just naturally more acidic than other soils. Like here we have a soil that is actually not acidic. We have a pretty basic soil here. In other areas like the tropics, you can have a really acidic soil. And this pH, this natural soil pH, affect bioavailability of all these elements which you see here. I put here the charge of the elements in the environment in soil to the left to see if we see any common rules. And I don't really see it, although I said earlier, well, in general, you can say cations tend to become more bioavailable at a lower pH, and anions tend to become more bioavailable at a higher pH, but that's not universally true. So there is more, it's more complex than that. pH is just one factor. But in any case, what you see here is that pH affects the bioavailability of basically all essential minerals, so that's good to realize. And of course, you want your plant to have access to all of them, because if the plant is deficient in one, then it's already wrong. Then that's going to limit plant growth. They have to have enough of every one of them. So if you would have to choose one pH that is kind of a best compromise to make everything sufficiently bioavailable, then you would end up somewhere here in this range, somewhere here between 5.5 and 6. That is a pH that is acidic, but the plant is okay. You don't want to make it so acidic that the plant is starting to get stressed. And at this pH, all of the essential elements are reasonably bioavailable. So that's typically what people give a plant. If you can control the pH, if you give a plant artificial medium, agar medium, nutrient solution, you often have it between 5.5 and 6. And that is because that's good for bioavailability. So you can see it can be quite extreme, the effect of pH. And if you think here we are at, well, what are we here, 7.5? So at 7.5, you can see zinc is likely deficient here. Copper, could be. Boron, manganese, wow. Phosphorus, big problem. So those things are good to realize. So depending on the soil type, you could use this as a guideline which elements could be limiting. So those would be something to focus on. You could either provide the elements at higher levels, or you could try to manipulate the pH yourself. Sometimes people, for instance, put lime into acidic soils to increase the pH. Or you could treat soils with a mild acid, like citric acid or something. And this is not only important for just plant growth, but it's also important if you want plants to accumulate enough of certain elements, or as much as possible of certain elements. If you think of nutritional value of crops, in some areas of the world, the certain essential nutrients for the consumer are really low in the crops because they're low in the soil or not bioavailable in the soil, and then the general crops in that country could be really low in certain elements like zinc and also selenium. So then the people who live there and also the livestock who feed on that forage from that area can get deficient. And in Western countries, that is more of a problem for livestock. People can take supplements, but in developing countries, they don't have access to supplements, and then you can have gigantic effects, and that's called the hidden hunger by the World Health Organization. So you have really billions of people in the world who are deficient in trace elements. And if you could boost crop levels of these trace elements, you could do a lot of good in the world. Yeah?
Would those trace elements collect in the grain, or would they have to eat these
That's a great question. To what extent would these trace elements end up in the grain? And remember that the grains, reproductive tissues, they are fed by the phloem. So that it depends on the mobility of the elements in the phloem. So some will end up in the grain and some will not. If they do not, then they would have to eat the whole plant. In the case of people, I don't know if you can get people to eat the whole corn or wheat plants, probably not, but animals can. So in that case, you could try to somehow genetically manipulate the plants to export more to the reproductive tissues, and people have done that. And things like vitamin also, people have genetically engineered to be produced in the grains, the golden rice, whereas normally they would not produce those things in the grains. So that's where genetic engineering can really help. Yeah. And also phytoremediation is another application of plants where you would want maximum accumulation. And then you could help the plants a little bit accumulate more by maybe manipulating the pH. So pH influences mineral bioavailability. Many cations are more bioavailable at a lower pH, and that is because the H+ concentration goes up when the pH goes down, because pH is defined by the H+ concentration. It's the -10 log of the H+ concentration. So the lower the pH, the higher the H+, and all that H+ is going to replace the cations from the soil cation exchange sites. And there's a similar thing going on for anions, but it's less important. So you also have anion binding sites which are positively charged on soil and organic matter. And if you have a higher pH, that means you have more OH minus, and that can also replace those from the positively charged soil binding sites. So that's why anions, as a general rule, tend to be more bioavailable at a higher pH. So we're going to have a look a little bit more at uptake, but first, just quickly, before we go back to the rhizosphere, let's have a quick look at the uptake process. So the first step of uptake into the root involves uptake through the plasma membrane into the symplast of the root. So that's similar to what you saw earlier for water. And this can happen anywhere in the cortex. So that could happen at the level of the root hair, which protrudes and increases the surface area of the root. So that would be here, this blue line, where our mineral would pass the plasma membrane by means of a protein transporter, which we will look at later. And now it's in the symplast, and once it's in the symplast, it can freely move through the cortex via plasmodesmata until it bumps, or actually it can get right into the endodermis as well, because they're all connected. If it would want to then get into the xylem, it would have to pass a membrane to get into the apoplast of the xylem. Another route that a mineral could take is they could follow the apoplastic route, which is shown pink, because cell walls are not any barrier for minerals. They're small enough so they can just diffuse with the water through the cellulose and lignin fibers all the way through the cortex, but then it would bump into the endodermis, the Casparian strip, which is impermeable for water and for anything dissolved in that water. So there it would bump into a barrier, and if it would then want to proceed, it would have to pass into the symplast, pass a membrane protein, and then it's in the symplast of the endodermis, and then again, if it would want to continue up to the shoot, it would have to pass a second membrane to get into the apoplast of the xylem. So here you see the functionality of the Casparian strip. It gives the plant control. 2 membrane steps are needed to get into the xylem. So control to keep out unwanted minerals, maybe cadmium 2+ would want to get in, saying I'm copper, but the plant says no, you stay out. And if the plant has done its best to acquire a lot of copper because it needs that, it doesn't want to leak it back out into the soil, so it wants to keep it in. So that's a good thing to have that control. But the cortex, just the cell walls in the cortex, anything can just diffuse in and out freely. It's also called the apparent free space. So that's just slushing about. So membrane steps for minerals to get across membranes involves proteins. Anything that is charged needs a protein to get across the membrane. And that includes even H+, which is the tiniest of solutes. It needs a protein to get across the membrane, because that hydrophobic interior of membranes repels anything charged. It's just way too polar. So keep that in mind. For minerals to get across membranes, you need a protein. And then usually if you want to go against the concentration gradients, which would mean from the soil into the plant, you go against the concentration gradients. The concentration is higher in the plant than outside. You want to kind of pump it up the hill. You have to put some energy into making this happen. So ATP is typically needed to move, to concentrate things in the root as compared to the soil. And that can be either directly or indirectly, as we will see when we look at these membrane transporter proteins. So some use ATP directly and some use it indirectly. But in general, energy is needed to take up minerals. And that energy needs to be produced by respiration mostly. So the respiration helps the plant produce ATP to take up minerals, and then the uptake of minerals is in turn needed to take up water via osmosis. You need the solutes to get the water in. So it all hangs together. And every mineral has its own transporter protein. So the membranes are full of different transporter proteins, and each transport one specific mineral. And sometimes you can even have 2 different transporters for one and the same mineral. like K plus uptake into the cell may have 2 or even more K plus transporters with different properties. So some work at a low concentration, some work at a higher concentration. So just imagine how many transporters there must be. I think it's 10% of the genome, of the genes, are involved in some sort of transport process. And there are also different transporters in different organelles. And you can also have different transporters in roots than in shoots, so different tissue types, or different in the vasculature versus in the epidermis. So lots of different transporters. And they're all regulated, and it must all be sensed. So in every different compartment, in every different cell type, continuously, plants must sense how much do I have of each of these elements. And then it must respond to that. So if there's a lot, it has to downregulate further uptake. If there's too little, it has to upregulate further uptake. And this all has to go on all the time. So it's pretty amazing if you think about it. So that was a little bit of a preview of how things get into the plant in the first place. And then now we're moving back out into the rhizosphere with a couple of other factors that affect uptake, particularly things that plants can do to manipulate their rhizosphere. You already saw they pump out H+, and that's reiterated here. So they excrete protons into their rhizosphere, the root zone. That decreases the pH, increases the H+ concentration, and that replaces other cations on cation exchange sites, and that makes those other cations more bioavailable so they can be taken up by the plants by means of those transporters that I just mentioned. Something else that plants do, particularly for metals, they excrete chelators. And the word chelator comes from claw. It's a Greek word for claw. It's a molecule that binds a metal typically. Although chelator is something that binds anything, but the word is usually used for metals. So plants can send out these chelators into their rhizosphere, and they can find the metals, bind them, bring them into solution. So they can wrestle them away maybe from the soil, which is holding on to those metals too, bring them into solution, and then they can reach the root, and the metal can then be released from the chelator, or sometimes the whole complex is taken up, chelator plus metal, and then a plant has its metal. So that's another mechanism that plants have evolved to acquire metals particularly. So metal acquisition is something that may limit plant growth more than CO2 acquisition. So plants tend to splurge with carbon. That's not so hard for them to acquire. There's always some carbon around. But metals, they're very frugal with their metals. They try to remobilize it from leaves as they senesce and are dropped. They really value metals. So another thing that plants can do is they can strike a bargain with microbes, bacteria and fungi that live in their rhizosphere, and that can help them take up minerals as well as water, by the way. This can include both metals but also things like phosphorus, so nutrients in general. And as an example, I'm going to show you some examples of iron uptake, which is particularly unbioavailable or bio-unavailable. So you need a strategy to take up iron, otherwise you're not going to get enough as a plant. And plants do both of these, actually all three of these things to take up iron. So they acidify their rhizosphere, that makes iron more bioavailable because it's a cation. They excrete chelators that bring iron into solution, so it makes it available. And plants have symbioses with microbes that can make iron more available. All of these things. So for iron, there are basically two ways in which plants use chelators to acquire them, and that's called strategy 1, which is most plants, and strategy 2, which is grasses. Strategy 1 uses phenolic compounds secreted by the roots, and sometimes these phenolic compounds may also be excreted by microbial organisms in the rhizosphere. In any case, phenolic, what that means is they're compounds with a ring in them. So also called aromatic compounds. So these concentrations of these kinds of molecules are there from biological origin, whether it's plants or microbes. They are correlated with the availability of iron. So we know for sure that some of those are produced by plants. They act as chelators, so they can bind metals, and particularly iron. And they bind it in the form of iron 3+, which is the prevalent form in most soils. So when there's oxygen present, the most oxidized form. They can bring it into solution, and then it can diffuse to the root surface. And then it can reach this enzyme here, which is a ferric reductase. You see it's an enzyme because the name ends in ASE. And the name of the enzyme tells you what it does. So it reduces ferric iron. And ferric iron is iron 3+. It reduces it into iron 2+, which is also called ferrous iron. And so reducing means it adds an electron. So the ferric iron 3+ gets reduced, it gets an electron, and it turns into iron 2+. And the enzyme that does this, the ferric reductase, uses NADPH, remember that guy, as the source of the electron. So then the NADPH turns into NADP+. So remember I was saying NADPH and NADH, those are molecules you see a lot being used by cells wherever redox reactions are needed, where electrons are needed to be given or taken. They can be used. So now we have iron 2+, after this has happened. Then that iron 2+ can be taken up by a membrane transporter protein into the cell, and then it can be further used and moved around. And this iron 3+ wouldn't be in solution if not for a chelator, which is just this phenolic compounds just hanging out in the rhizosphere produced by the plant or produced by other organisms. And they actually release the iron from the soil. Iron tends to form complexes, insoluble complexes with soil, like hydroxides. But the chelator is able to withdraw this iron from the soil particle, bring it into solution. And then it's mobile, so then it can actually reach this ferric reductase. So it's a 1-2. It's the two proteins working together. One reduces the iron, and then the other one takes it up through the membrane. And then we also see here this proton pump, which I mentioned earlier. Plants pump continuously H+ protons into their apoplast, so through their plasma membrane. And they do that at the expense of ATP, so that's the other guy that's really important in metabolism. It's a currency of energy. So to pump H+ against its concentration gradients, because the H+ concentration is much higher in the apoplast than in the cytosol, at least 100-fold higher. So to pump that H+ out of the cell continuously, ATP needs to be used up to make that happen. Yeah, question?
Why is it, even if you're saying they're getting it, why is it not able to take it up as the 3+ charge, but has to be a 2+?
Why is the iron not just taken up as iron 3+? Yeah, good point. I don't know. Maybe we should tell the plant. Why are you doing that? I guess there must be some reason for it. It's used in the plant usually as 2+, so maybe if it has to reduce it somewhere, it might as well do it right at the beginning. But, nah. Actually, grasses do take it up as 3+, as you will see in the next slide. But for some reason, other plants don't. Maybe it's easier this way?
Grasses take it up
as a complex 3+, yeah. As I'll show you, strategy 2. Okay, so there are phenolic compounds, they bring it into solution, they bring it to the root surface, it's reduced, iron 3+ to 2+. In the process, it's then released from the phenolic compound, and it can then be imported into the root through the membrane by a membrane protein. It says here carrier. Technically, it's not really a carrier. But anyway, it's a membrane protein that takes up iron. Okay, so here you see a close-up of the same thing. Yeah?
Why does the plant allow H+ iron, or H+ to take up the iron, or is that just a separate thing?
It's the pumping out of the H+ is separate from the other processes, but it also helps make the iron more loosely bound to the soil, because if you have other cations that kind of are competing with it for the soil, negative soil groups, there's more likeliness that it will come off, but it's independent. Yeah. Okay, well, I see that it's time, so we'll keep strategy 2 for next time. about?
Can you tell me about echo?
Echo, yeah.
Yeah, because